What if I told you that we can find out what you ate from a poop sample this big? Hi, my name is Kyla Geronimo-Anctil, and this summer I worked with Callie Wise and Dr. Katrina Lohan in the SERC Coastal Disease Ecology Lab, or CDE. For my project, I looked at the diet and parasites of river otters through their poop. But there's one big problem. There is biological material literally everywhere, and my project is really sensitive to this type of contamination. Contamination is what things get mixed that shouldn't really mix. Contamination can send false messages about what otters ate when things that they didn't eat get mixed in their poop. We want to make sure that the result that came from one sample actually came from that sample and not the one right next to it. To minimize this, we use aseptic techniques, which are a collection of different practices to prevent contamination of samples by removing non-target DNA. Aseptic techniques are important because contamination can lead to inaccurate results, and in turn, we draw less accurate conclusions from our data. And because we are looking at such small samples in my project, there's a very high chance of contamination. Aside from wearing gloves, because even your skin is a biological material, we also heavily clean our workspace and our tools. We have to be careful about how we do this though, because we use a flame to sanitize our tools and bleach to sanitize our workspaces. But if the bleach mixes with something like rubbing alcohol, that makes chloroform, which is a super toxic gas. Thinking about it this way, you have a friend visiting you who is very allergic to pineapple. You want to give them some food, but you just cut a pineapple on your counter. You are likely going to clean the counter and any tools that might have touched the pineapple, even if you're not 100% sure that you used them, because you want to be sure that there is no pineapple on the food you give your friend. It's a similar idea when we clean our tools and workspace. Every time that we clean a tube with a sample or interact with the sample in any way, we need to make sure that our tools are clean and sterile before and after this interaction. Remember when I said that biological material is everywhere? Something interesting is that it affects how we open tubes and other containers. We have to make sure that we do not touch any part of the inside of our containers, or else we may contaminate our samples. One way that we can do this is by using one hand. Notice that no part of my finger touches the inside of the lid. We also have to be careful to always keep the lids on our containers unless we are immediately using them. There's biological material just floating around in the air, and the longer that our lids are open or off, the higher the risk of contamination to our samples or our other containers. Using aseptic techniques, we can be more confident that each result came from a unique sample and learn about the diets that poop tells us about.